So basically, my intention was to yesterday introduce the models that are more coming from the material science point of view. And today I wanted to introduce two models that comes from biomathematics. And first one is the model which I'm currently working on. It's motivated from tissue engineering. And the second one is something related to modeling of diabetic kidney disease. So I'll give a brief introduction of both of these two models. So to begin with, so basically nowadays there is a huge research going on to develop artificial tissues which can replace, so sometimes it is not possible to find a donor whose tissues you can use when you have a damaged tissues in your body, which you could have, one can damage by, doing heavy sports, or if they're doing heavy physical activity, or also aging can also cause tissue damages in a person's body. For example, if you're too sporty, then in that case, sometimes the cartilages, they get damaged, because of the rotation or twist in your knee. And cartilage is sort of like a thin filament in your knee, on top of which, both of your upper part of your thigh and the lower part of your leg, they sort of slide against one another. So sometimes that gets damaged, and in some situations, you have to cut down the damaged tissues and then replace it either by some tissues from your body, from your own body, or from some donor. And nowadays, people also trying to replace it by doing some in vitro tissue replacement. That means you prepare, or scientists are making artificial tissues. They are called as scaffold structures. And you basically do the cell seeding in these scaffold structures, and you replace the damaged tissues, so cutting down the damaged part, and then you replace the damaged tissues with the help of these scaffold structures. And the overall idea is to study these scaffold structures, that what would happen when you apply a certain load, then based on the load, how these scaffold structures, they react. Because once you, for example, if you replace the damaged cartilage by this artificial tissue, then it goes under twisting and turning and all that, so whether it can get damaged again. So the purpose behind these scaffold structures, so for example, here is the knee figure. So suppose if the cartilage in between the knee, if it is getting damaged, then you can replace it by these kind of scaffold structures or artificial tissues. And they can be visualized as a periodic structure. So they are composed of these microfibers. So these composites, or these structures are made by these microfibers. And inside these microfibers, you have some gel. So basically, it's a very pore elastic kind of medium. So the fibers are basically elastic, and the gel inside it is pore elastic. So overall, you have a very elastic medium such that it can, if you're replacing your cartilage with an artificial tissue, it should do the same thing what your actual cartilage does. And therefore, you need to have a very fluffy, or a tissue-like scaffold in your knee, for example. And if you look carefully, then this is also like a periodic structure. So you have microfibers, and inside these microfibers, you have hydrogels. And they are also periodically distributed all over the microfibers. And the overall idea to study this scaffold structure, as I said, that what would happen when you apply the load. So when you apply the load, how this overall these scaffold structures behave. So something we call orthotropic behavior of these scaffold structures. So orthotropic means when you are putting it in your body, how they are behaving under a certain pressure. So that's what we are trying to analyze by studying these scaffolds. And since they are microscopic structures, we can periodic structures, we can reduce the whole model into a micro-macro situation. So by doing the, first of all, we will consider the model at the micro scale, which will be composed of linear elasticity equation and then the equation for the hydrogels, complemented with some stress boundary conditions. And when we are doing the homogenization, we are basically trying to have an upscaled model where you can perform the numerical simulations by putting these loads as a parameter, that if you apply this much pressure, then overall how your scaffold structure would behave. So that's the two aim that we are currently focusing on. So basically, if you look at a very small micro level, so what you would see is that these scaffold structures, they look like this. And in between these, cubes or hollow structures, you have some hydrogels. And if you want to visualize in terms of a periodic structure, so these are basically your microfibers, which are these. And these hollow parts, that's where you have the hydrogels. So overall, you have a very elastic medium. So these microfibers, they are assumed to be elastic. And these hydrogels, they are assumed to be, also elastic, but since they are lying in the pore, we are saying that they are pore elastic. And the microfiber, these fibers, they are elastic in a way. So basically, we have, if you model everything, then we'll end up with basically a pore elastic model. So as we are saying that these microfibers, they are elastic. So basically, if you assume that the displacement of these microfibers are given by UF, then in that case, this UF will follow the linear elasticity equation, which is given by this, where mu F is the modulus of stress, and lambda is the Lamé's first parameter. And this is the general, the linear equation or general equation of elasticity for any elastic material. And we say that the hydrogels, they are pore elastic. So let the displacement of these hydrogels are denoted by UG. So G is for the gel. And we say that since they are hydrogels, so if you apply even a little bit pressure, then these hydrogels, they will move a little bit. So we are denoting the velocity of these hydrogels by V, and P is the pressure. And since this model is pore elastic, you will also have an elasticity equation for the hydrogels as well. And on the boundary, we assume that the stresses are continuous. So the Cauchy stress tensor at the interfaces, so the stress tensor for the fiber part and the stress tensor for the hydrogel part, they are continuous. This is a standard assumption that we make. And the displacements are also assumed to be continuous. And there is no, that the hydrogels from one cell is not jumping into the another. So there is no mixture of hydrogels. So we are considering that at the boundary of these, microfibers, nothing is going from one cell to another cell. So that's our, global model. And then we did some non-dimensionalization and introducing the scaling parameter. Ultimately, we made these assumptions that omega is the given scaffold structure, and we denote we consider a unit cell Y, where we denote YF as that microfiber, and YG as the gel. And based on that, as we see in the homogenization theory, you say that everything is covered in the translated versions of the cell Y. So then in that case, our YF, the microfibers in the entire scaffold, will be the union of these microfibers all over the scaffold, and hydrogels will again be the union of all these hydrogels in the entire scaffold. And the boundaries between the hydrogel and the microfibers are denoted by gamma epsilon. So then if we say that W epsilon is the deformation for the fiber, and U epsilon as the deformation for the gel part, then in that case our entire general, yes. Why? Oh, sorry, this I is alpha basically. I messed it up, yeah. Yeah, I was changing the notation, so I forgot to change this I. So it should be alpha. So union of all the fiber parts, and then union of all the gel parts. Yes, thank you. So basically, this is how our entire model will look like at the microscopic level. So that is our linear elasticity equation for the microfibers. And this is the pore elastic system for the gel structure. And that's the interface condition. When we were doing the modeling, we were not sure with the scaling parameters. So sometimes you see that diffusion reaction equations, in some of the papers they have epsilon square in the diffusive term, sometimes they have epsilon, sometimes they have epsilon to the power k, where k is running between some ranges. So here we said that we started with epsilon to the power beta, epsilon to the power gamma. So for different values of beta and gamma, you'll end up with a different model at the upscale level. So if your beta is 0, gamma is 0, you'll have a different model. If your beta is 2, gamma is 0, then you'll end up with different model. So basically what we wanted to do, we wanted to have a model where we can at least perform the numerical simulation, where we can obtain the homogenized model using two-scale convergence. So we played with these parameters. So these many cases can arise. So either both the parameters are 0, or you can have gamma as 0 and beta as 2, that is assuming here epsilon to the power 0 and here assuming epsilon square. Assuming gamma as 2, beta as 0, that means there is no epsilon here, but we'll have epsilon square here. Your gamma is 0, but beta will run between 0 to 2. Your gamma will run between 0 to 2 and beta is 0, and when both will run between 0 to 2. So there are several possibilities, scaling possibilities that you can play with, and for every value of the scaling parameter, you'll end up with a different model. So there is a very nice paper by Michel Bohm and Malte Peter from 2008, where they have addressed these issues that if you have different scaling parameters in your diffusion reaction equations, you'll end up with different type of upscaled models. It's a very classical paper, and I think people working on homogenization theory should have a look at that. Now, so for us, the most suitable scaling was taking gamma equals to 0 and beta equals to 2. So we verified most of these cases, and it turns out that this one was something that we wanted to study. All right. So then the required model, where we considered epsilon square and epsilon to the power 0 in our model. And now, so since we have the continuity of the displacement, we introduced a capital U epsilon, where when we are in the microfiber part, we'll denote it by W epsilon. When we are in the gel part, we'll denote it by U epsilon. And then we can define the weak solution in this fashion, where we are looking for the solution in this space, where the time derivative is in the dual space. All right, that's the standard weak formulation setting. And then by usual energy methods and Galerkin's approximation, we can be able to obtain the existence of solution, and at the same time, this type of a priori estimate. Yes, this capital U is actually accounting for both fiber and gel. Yeah. So the fibers, are outside the gel. So fibers are outside the gel. So those are the Yes. So yeah, exactly. Yeah, that's what I'm saying here. All right. So now we can be able to obtain an a priori estimates like this, and then we can do our usual extension, and then we know that if you have the extension, then you can find out the two-scale limit. So basically, the gradient of U epsilon will converge to this limit. I think you have seen this through the workshop at least 4 or 5 times. and all these U epsilon, W epsilon will converge to their usual 2-scale limit. All right? And now we have the limit. Basically, now we can pass through the limits in each one of these equations. So we'll first consider the microfiber equation, then we'll consider the hydrogel equation, and then we'll consider the pressure equation where you have del P del T for the hydrogels. So in case of microfiber equation, we'll consider the test function in this fashion. Then we multiply by the test function, and using the 2-scale convergence definition, our stress tensor basically will converge to a limit like this, where capital C is the 4th order tensor, and hence we have the tensor product here. And you can visualize in this way that, so we can visualize in this way that E of W epsilon is basically 1/2 gradient of W epsilon plus gradient of W epsilon transpose. So that's how we define the Cauchy stress tensor. And when we are passing the 2-scale limit as epsilon going to 0, so then in that case, this will converge to 1/2 gradient of W plus gradient of Y W1, and then gradient of W transpose plus gradient of Y W1 transpose. And therefore, here we are obtaining 4 terms actually. So the stress tensor will actually converge to a stress tensor which is given by EW plus EW1, where EW is accounting for the x part gradient of W, and EW1 is corresponding to that gradient of W1 with respect to Y. And since it is defined in this fashion, if you break it, then you'll basically end up with this expression here. All right. Next, doing the similar technique, we can be able to obtain the upscaled elasticity equation for the hydrogels. It will look exactly the similar way as for the microfibers. And the pressure in hydrogel, the third equation, will look somewhat like this. And of course, we'll have our usual boundary conditions, which can also be obtained by 2-scale convergence. And if we want to calculate each parameter individually, so I mean, the calculation is a little bit messy. So we have to solve a cell problem like this. So if I go back to our homogenized model, so when we consider phi 0 equals to 0, then in that case, we'll have a cell problem. You can solve the cell problem to obtain these effective parameters. So we can obtain the effective Cauchy stress tensor. We can, if you charge the gradient on it, then it would look like this. So the same elasticity equation which you had earlier at the microscopic level. Similarly, we can calculate the effective velocity on the hydrogel part, and we can also calculate this Darcy law for when we are doing the upscaling for this equation. If I go back for this type of equation. So this is purely doing integral here and there and obtaining the way usually we obtain the Darcy's law from these Stokes equations. And as I was saying, we can make that phi 0 equals to 0 for the elasticity equation of the microfibers and elasticity equation for the hydrogel. And then we'll obtain 2 different cell problems. By solving these cell problems, we can be able to determine our tensors D and C. And in the representative cell Y, this will be our stress condition, the continuity of the stress condition, and that's the continuity of the displacement. So the continuity of the displacement is assumed in a way, a very classical theory, which we know that if you have let's say a solid plate, and if you have a fluid touching it, then if this plate moves, then the fluid which is touching the plate, it will also move with the same velocity. Hence, you have the continuity of the displacement assumed here. And that would still holds in the upscaled equation as well. Okay, so this is a very recent work that I did with one of my colleagues from University of Bremen. And in fact, the model which we considered here is also not that old. So we adapted this model from one of the papers by Helen Byrne from University of Oxford. And she has published this work in last year or 2017 or something. So they are engineers, and they did the upscaling more from the asymptotic expansion point of view, whereas we were interested in more like two-scale convergence and if possible, then use periodic unfolding. But fortunately, we didn't need unfolding. We were managed to obtain the upscaled model using two-scale convergence itself. Yes, we can go. So, so this is overall the, let's say, I can treat this as a scaffold structure. So suppose this is the scaffold structure where you have the microfibers, and inside those square microfibers, you have the hydrogels. And these microfibers, they are distributed in a periodic way. So basically, we are trying to analyze the global behavior of this microfiber when you apply load onto it. So we are interested in the orthotropic, means how it behaves. No, not orthotropic. Orthotropic, I'm lacking the word. So basically, when we are replacing it in our body, I mean, how it behaves. So exactly. So my periodic cell looks like this. Yeah, I have the, yes. So my periodic cell looks like this. Yes. You mean here, what does the epsilon mean? Because first of all, these displacements and displacements for the microfibers and for the hydrogels, I mean, we want to analyze their global behavior, right? How they are behaving. So in order to do that, we first of all reduce the entire model at the microscopic level. And then you sort of do the... Which coefficients? The C's? No, they are. I mean, that's basically your the fourth order tensor in your Cauchy stress tensor, basically. Yes. So basically here, it does. Yeah, yeah. The size does go to 0 because you cannot have a very big cells where you have the fibers and hydrogels. I mean, ultimately, you're trying to consider a smooth structure which can sort of floats when you are replacing, not floats, but behaves like a pore elastic medium. So we are ultimately sending the epsilon to 0 so that you can have a homogeneous scaffold structure. Yes, yes, yes. That's the only case where I can pass to the limit. Yes.
Okay, so it's not clear that your limit has any implication for an engineer on how to construct this
Yes. I mean, that's what I said. I mean, multiple possibilities can arise if you play with the scaling parameters, and which one those engineers are looking. It's just that we have to do trial and error and see if they are satisfied with it or not. Yeah. Any other questions motivated from this model? Shall we go to coffee break and then continue the next model? That would be good. Huh? No, I'm not tired. I'm just saying. Okay, perfect. Then let's continue. Okay. Okay. All right. So now I don't want to eat up the coffee time, so that's why I said. Okay, perfect. So let me continue then. So my next model is basically about diabetic kidney disease. This was the project that was given to my postdoc supervisor from one of the pharmaceutical companies from Sweden, AstraZeneca Pharmaceuticals, and their project was divided into two parts. So first of all, they still have this drug for diabetic kidney disease. So the overall idea is that once you have diabetes, you cannot reverse that. So diabetes is incurable. It's a bad news. Now, if you have diabetes, then it's sort of like a snowball effect. So diabetes sort of brings high blood pressure, and at the same time, it will damage your kidney. So if someone has diabetes, their kidney is going to get damaged, for sure, and they need dialysis sooner or later, maybe 10 years down the line or 15 years down the line. So there is no reverse. So, you cannot cure that. So this company, they developed a drug which can not cure diabetes or which cannot cure the kidney disease, but it can slow down the process. So if one kidney is supposed to get damaged in 10 years, then it might slow down and it might last for 15 to 20 years. Eventually it will get damaged. It's just that the process will be slower. So this company, they came up with this drug, and they did some experiments on patients who has several types of kidney diseases. So you might detect it at the very early stage. So that means that's called as chronic kidney disease stage 1. You might detect it at a very later stage where 50% of your kidney is damaged. So that's called as chronic kidney disease, maybe stage 3. And if someone is already close to getting kidney transplant or dialysis, that chronic kidney disease stage 4, that's the last stage. So they hired different patients and they gave them the same drug to these different patients, and they came up with experimental results. And it is also well known in this nephrology community that our kidney has very, non-uniform distribution of nephrons. So you can visualize kidney also as a heterogeneous medium where every nephron is of different size. But when they did the experiments, they sort of assumed that each nephron is doing the same function. So like assuming a homogeneous behavior. So that was the first assumption they had. And the second thing which they did is they collected all these experimental results. But in order to get the approval from FDA, so when you want to launch a new drug, you have to get the approval from FDA. And to convince FDA, they need some mathematical model or numerical simulations, which they can show that, okay, if you give the drug at this stage, it will do this thing. If you give the drug at this stage, it will do this thing. So basically, a deterministic model where you can feed in some parameter and that will give you some kind of predictions, which should match with the experimental results. So if it is not matching with the experimental results, then the model is again not useful. So that's where I came in. So we developed a mathematical model that can predict if someone has diabetes, then without the treatment, how long it will take to reach up to a certain level of kidney disease. So whether that person will go to stage 1 in 10 years or stage 2 in 15 years or stage 3 in 20 years, that can be predicted with this model. And when we had this model, then that's when we jumped into the second question, that if the distribution of nephrons are not uniform, then they are also not filtering uniformly. So it's very vital to consider one of the nephrons, analyze it, and then you do the upscaling to the whole kidney. So you can visualize your kidney as also a heterogeneous medium. So first of all, I'm going to introduce the basic model where we don't have any heterogeneities or anything like that. If I'm getting too much biological, please stop me, and then I'll jump to the next mathematical equations or next mathematical results. So it might get a little bit too biological. So to give you the motivation, why kidney? What is so interesting about kidney? So kidney is actually one of the major organs in our body. And it controls, first of all, it filters all the toxic product from our body. Like it works as an excretory machine. It also maintains a very tight level of sodium and water. So imagine the days when you don't drink water, and if the person uses washroom, the urine color is yellowish. It's mainly because your kidney senses that today I didn't drink a lot of water. So I have to keep the water in my body to keep me hydrated. So your kidney excrete more sodium. Hence, the color of urine is yellow. But the days when you drink a lot of water, the urine remains whitish, mainly because the level of water and sodium in your body is same and the excess water is being excreted. So water is colorless, hence the color of the urine is whitish. So that's how kidney controls very tightly sodium and water level. And it's really amazing that how it functions. I mean, whenever you eat chips, your kidney senses that, oh, my salt level is up. Give me something to drink. So you immediately crave for water or cold drink or whatever it is. I mean, it acts like a switch. And therefore, it is quite important. It also regulates the blood pressure. Because if in your body, if sodium is more, then what would happen is, in order to keep the same sodium level, your kidney will try to keep the same water level. So if you have high sodium level, you will also have high water level. So having high water level means basically you have high blood in your body. And since our body is not like a balloon, so it cannot grow, so what will happen is it will shoot your blood pressure. It will immediately shoot your blood pressure. That is why people who have blood pressure, they tend to eat more. They crave for food because they always try to keep the same sodium level, and since the water level is anyways high, so they try to keep the sodium level high, and when their sodium level is high, their water level is high. So it's sort of like again a snowball effect. So people who crave for more sodium or who are eating more, I mean, you might say that you'll get blood pressure or you'll get some kind of heart issues, mainly because they are supplying too much sodium in their body. And if they are supplying too much sodium, their water level in the body will also go up. And that is also tightly controlled by kidney. Now the problem is, if your water level is going up, that means your blood pressure is going up. And if the blood pressure is going up, then all these arteries and veins, they have, how to say, a fixed diameter. So if you have more pressure, so what would happen is either these vessels or capillaries, they will grow in order to accommodate that pressure, or they have to deal with it. And imagine all the veins and arteries that is going to the heart. If the pressure is, when the pressure is more, it's more everywhere. So that means the veins and arteries that are going to the heart or coming out of the heart, they will have more pressure. So in a way, kidney is also controlling your cardiovascular functions. So it works as a very strong organ in our body. It controls our sugar level, it controls our sodium level, it controls our blood pressure, it controls our cardiovascular functions, and hence it is important to study. I mean, people always say that take care of your heart, they neglect kidney. But most importantly, you should always take care of your kidney, you will be indirectly taking care of your heart. All right? So it's very important. Now, and each kidney, they have this kind of structure. So this is called as, so kidney is composed of these kind of structures. They are called as nephrons. And nephrons are nothing but a bunch of capillaries going in this way, where the blood that enters through these capillaries, they filter some stuff like sodium, glucose, water, and then some of the stuff that gets reabsorbed, and rest of the things are excreted through the kidney. So basically, nephrons are the functional unit of kidney. We may have studied that a long time ago in our high school. And a healthy human has almost 2 million nephrons. Now, yes. So how do we measure that whether our kidney is functioning okay or not? So whenever a person who feels that they have some kind of issues with their kidney, they usually go to the doctor. And what the doctor does, the doctor measures something called glomerular filtration rate. So in the previous figure, this structure is called as glomerular, where the filtration happens. And the doctor usually measures that how much this glomerular is filtering. So there is a certain range. So it must filter 9 milliliter per minute blood. So if it is filtering less than that, that means the kidney is not functioning properly. Our kidney filters 180 liter of fluid every day. So it is a constant machine that is working ever since we are born. and it is doing the same filtration process again and again. So basically, this is the parameter that doctors measure, glomerular filtration rate, by the help of which we say that whether the kidney is functioning properly or not. And usually they take the blood sample and they test for this protein. Am I talking too much biology? So please stop me if it is too much biology. Is it okay? Huh? All right. So basically, what they do is this is a protein that they measure, and this protein is a very big molecule. So they are very big molecules. So if someone's kidney is damaged, then in that case they filter these molecules in the urine. But if the kidney is healthy, you don't filter these molecules. So they usually measure these molecules in our blood, and if these molecules are low in our blood, that means it is getting excreted from somewhere. So it is excreted from our kidney, and hence they say that the kidney is damaged. But if this creatinine level is within the range, then our kidney is functioning well. So usually if our filtration rate is greater than 90 milliliter per minute, then we say that our kidney is healthy. Otherwise, we categorize into these 4 categories. Chronic kidney disease 1, stage 2, stage 3, stage 4. And once a person crosses stage 4, then it's a kidney failure situation. Then that person needs dialysis or kidney transplant. Now comes the mathematics, basically how we did modeling. So basically, this is our glomerular, and that's where the blood is coming in. So through this artery or capillary, the blood is coming inside the glomerular, and when the blood is getting filtered, so these are the filters. So that's where the blood enters through these filters, and they sort of filter our excretory products like sodium, glucose, and other stuff. So basically, we have some kind of pressure. So when the blood is entering, it is entering with some pressure. We call it as this hydrostatic pressure is called as glomerular pressure. And since this capillary is very elastic, there will be a an opposite reaction kind of thing. So that opposite reaction is called as oncotic pressure and Bowman space pressure. So if the flow is in this direction, then there will be two opposite pressure acting in the opposite direction. So they are called as oncotic pressure and Bowman space pressure. So the net pressure will be given by the difference of the hydrostatic pressure that is entering here minus these two negative pressures. So whatever you have entering, the hydrostatic pressure minus the counteractive pressure, so that will be the net pressure if you take the difference, which is given in this way, and then you multiply by a scaling factor, Kf, and that Kf multiplied by this net pressure is called as the filtration rate for 1 nephron. So filtration rate for 1 nephron is given in this fashion, and if you want to know how much or how well our kidney is filtering, you multiply by the total number of nephrons. So we first find out how much, what is the filtration rate for one nephron, and then you multiply by the total number of nephrons to know the filtration rate for the entire kidney. All right, and as I was saying that if we have too much sodium level in our body, then we will have too much water level in our body, and that too much water level and sodium level will again constitute to our cardiac output. So it will basically affect our cardiovascular functions. Hence, higher pressure will result in bad cardiovascular functions or sometimes cardiac arrest or heart attacks. So having higher sodium level in our body is not a good sign. So that's why people say do dieting and avoid chips or all the salty products, mainly because through these salty products we are actually shooting up our blood pressure. So our goal was how to model mathematically the filtration rate, the glomerular pressure, the sodium, how much sodium we are excreting, how much water we are excreting from our body, and how we can model the blood pressure in our body. So all these things can be modeled by this relation. So the hydrostatic pressure is given by the given by the total blood pressure or mean arterial pressure minus the blood flow. And single nephron filtration rate is given by Kf times the net filtration pressure. Then the total filtration rate is given by number of nephrons times single nephron filtration rate, and how much glucose we are filtering. So this is the concentration of glucose, and how much we are filtering from one nephron can be given by multiplying the filtration rate for one nephron times the concentration of glucose. And after the filtration, we also have a channel in our kidney where you reabsorb all the glucose. So this parameter will give you how much glucose we are reabsorbing, and therefore this parameter will give you how much glucose we are excreting. So our kidney also reabsorbs some glucose, but there is a cap after which it cannot reabsorb anymore, and therefore whatever left, it will get excreted. So if we have too much glucose in our body, our kidney can reabsorb up to a certain amount, and after which it will excrete the rest of it. So if it is excreting, that means we have already too much sugar in our body, because kidney has a capacity after which it cannot reabsorb anymore. So it will start excreting that, and if it is excreting glucose from the urine, that's a sign that the kidney is damaged. That's why you may have heard that some people say that if there are ants collecting to the urine, that means the urine has glucose in it, and then it's a bad sign that the person may have diabetes. So that's the reason behind it, that our body cannot reabsorb all the glucose, and we have excess glucose, so we are excreting it. All right. And similarly, we can model the excretion of sodium, because we also need to get rid of sodium so that we don't have high blood pressure. And the sodium filtration can be modeled in this way. And the rate of change of concentration of the glucose and of the sodium can be given by these equations. So this is the net glucose in the body, and this is the net sodium in the body, and then how it is changing over time can be given by this system of ordinary differential equations. So I'm saying that diabetes actually a bad news for the kidney.
Okay, the other.
The other way, yes. So if no, yeah, I will show in a minute that if someone has high glucose level, so having diabetes means having high glucose level. So if someone has high glucose level, then how it will affect the functionality of the kidney. Yeah.
Also,
No. It's always other way. Other way. Okay. Yeah. So.
So homogenization is the other thing.
No. I do not get other way. Yeah. Don't worry. Are you with me up until here? Is it boring or okay? All right. So basically, what happens is so theoretically. Experimentally, what happens is when you have high sugar level in your body, that's what we mean by diabetes. Then sugar and sodium, I will show in 1 or 2 slides, that sugar and sodium, they sort of get reabsorbed in our body in 1-to-1 proportion. So if our kidney is reabsorbing 1 molecule of sodium as sugar, then it will also absorb 1 molecule of sodium. So whatever sugar is present in our body, if it is getting reabsorbed, the similar amount of sodium will also get reabsorbed. And this is a very recent theory that came into the picture, that sodium and glucose reabsorption are done into 1-to-1 fashion. So what would happen is, if you have high glucose level in your body, so all the glucose will get reabsorbed by the kidney, and therefore all the sodium you have present, that will also get reabsorbed by the kidney. So kidney not only filters, it also reabsorbs. It reabsorbs all the glucose, it reabsorbs all the sodium. Because if it is excreting all the glucose, then we'll just simply, have low glucose level. And if it goes beyond a certain level, then a person dies as well. I mean, having high glucose level is not a bad news, but having less glucose level in our body is actually very dangerous situation. I mean, if it goes beyond a certain limit, then the cells in your brain will start to swallow and the person might die. So the kidney, I mean, it works as such a nice mechanism that, again, that's what I'm saying. It works in such a nice way that it reabsorbs all the glucose so that you don't have low glucose level. That's what happened with these kids from Muzaffarpur and encephalitis, the disease where they ate all the lychees, and a lot of kids died in Muzaffarpur. Have you heard the news? Yeah, so the idea was that if you eat too much these lychees, then it brings down the sugar level, and if it is bringing down the sugar level, then it goes down to such a contagious level, then it actually becomes life-threatening. So having low sugar level is a very bad news. Having diabetes is a bad news, but having low sugar level is actually deadly. So kidney tries to keep that also in check. It reabsorbs all the sugar, but again there is a capacity that a kidney cannot reabsorb everything that you throw at kidney. So it will again excrete. So when it is doing the reabsorption of all the sugar, it will also reabsorb all the sodium in our body. Now if you already have too much glucose in your body, then that's a sign that you also have too much sodium in our body, because kidney will reabsorb sodium as well. And if you have too much sodium in your body, then your blood pressure will always be high. And if your blood pressure is always high, then the pressure through your arteries and veins will always be high. So all the veins and arteries that is going to the kidney, they will also experience high blood pressure. Yeah, then they will also, it will get much nicer, don't worry. So then all the veins and arteries that are going to the kidney, they will also experience high blood pressure. Now what would happen is the veins and arteries that are going to the kidney, they sort of start to increase in size. So in order to adapt that high pressure, either the length of the tubules has to increase or the diameter of the tubules has to increase so that they can adapt that high blood pressure. So the tubules, they increase in size. And if you check the kidney of a person who has diabetes, their kidney is bigger than the normal kidney. And it is very irregular shape. It's mainly because their kidney is trying to adapt the high blood pressure in the body. And that's the first sign that the kidney is about to get damaged. So that means if someone has diabetes, they will have high blood pressure. And the very first stage of this blood pressure, high blood pressure, is that the kidney will start to grow. But again, these are all elastic stuff, so they cannot grow forever. And therefore, after growing for 30% or 40%, your kidney will saturate. So it cannot grow forever. But since we are not controlling our diet, we are keep eating whatever we are eating, so the pressure will keep on increasing. I mean, it will remain that high pressure all the time. And over the time, these high pressures, they will start damaging those tubules. So it will slowly start damaging your nephrons. And what would happen is, over the time, your nephron functionality will start to decrease. Because suppose you have 10 nephrons, you have high pressure, then it damages the let's say 10th nephron. So now you have 9 nephrons doing the work of 10 nephrons. So it's like 9 nephrons are doing more work. So over the time, the 9th one will get damaged. Similarly, the 8 nephrons will do the work of 10 nephrons, so the 8th one will get damaged, and so on until all of them will get simply damaged. So it's like in an office, when one person leaves, if 9 persons are doing the job of 10 person, then they'll be a little bit frustrated. Then again, one leaves, then the 8 person are doing the job of 10 person. Then those 8 person will be a little more frustrated, and it will keep on going until the last person quits. So it's the same thing that happens with our kidney. And based on that, so basically so these equations, they actually model the several parameters in our body, like if you feed in the number of nephrons, the glucose intake, the sodium intake, the water intake. For example, we drink 2 liters of water every day, and certain other parameters. Then in that case, our model can predict the GFR, sodium excretion rate, blood pressure, cardiac output of a healthy human very precisely. So our model was able to capture the physiological effect for a healthy person if you feed in the parameters for a healthy person in our model. So actual system is very complicated. So we have like more than 100 equations with 100 different parameters. Here I have highlighted only a few important ones, like sodium reabsorption, glucose reabsorption, but the actual model is quite complicated. That's where all these parameter goes in. And this is the place where we modeled the physiological responses for healthy humans, and we also matched our data with the literature data, and it fits very well. So actually, our model was very successfully was able to capture the physiological effect for healthy humans. And then we sort of asked the question, what would happen when a person has diabetes? That means when you have high glucose level. So if you have high glucose level, as I said, our kidney will start to grow. So that's something called hyperfiltration. So our kidney will grow in size, but due to high pressure, as I was just saying, over the time, the kidney function will start to decrease because you are damaging the nephrons over the time. And over the time, the bigger molecules like proteins, they will start to excrete in the urine because your nephrons are getting damaged, so you are excreting bigger molecules. So that's how we describe the diabetic kidney disease, that over the time, the kidney functionality will decrease and the number of protein molecules in the urine will increase. So that's what we mean by diabetic kidney disease. And this effect we also modeled it successfully. So we first of all considered a rat model or a mice model. So this is the continuation. So now we are modeling the diabetes effect. What is that? Homogenization. Yeah, that's where I'm coming at. Yeah. So as I was saying that now we are going to model the diabetic effect. So we increase the glucose level in our model, and so basically what would happen is we considered this work of Levine, and there they have modeled the diabetic effect in mice. So we increase the glucose level from 85 milligram per deciliter to 556 milligram per deciliter. That's a very high glucose level in the body. And in their paper, they observed that initially the filtration rate would increase because you have bigger kidney, but over the time, since the kidney will get damaged, the filtration rate will also start to decrease. So this effect our model can also be able to capture. So at 8 weeks when we gave them diabetes, the filtration rate immediately increased because when you gave them diabetes, that means if they have high sugar level, then they will have high sodium level. But to bring the sodium level back to normal, the kidney will increase the blood pressure, and therefore your overall filtration rate will increase. But again, over the time, it will damage the kidney, and therefore your filtration rate would decrease. And I think I'm getting into too much biology, so I will skip few slides, if that's okay, because yeah. So now, when we presented this work was already published last year in Nature, Nature's Systems Biology, and after the publication of this work, this company, they gave us another funding for to hire one PhD student and one more postdoc, but it's just that my contract was exhausting, so it could not be renewed. But we have a PhD student working on this model. So our next task was to treat kidney itself as a heterogeneous medium, because it's been found that the nephrons, they are not of same size. So one nephron is bigger than another one, and they also have different filtration rate from when you go from one nephron to another nephron, and so on. So based on that, this distribution of nephrons is also not very uniform. I mean, it's not uniformly distributed. So we sort of consider to treat it as a periodically distributed nephron system in our kidney. Yes. Multiplication of what? This one? There is a dot here, actually. Yeah. Yes. So I sort of assume people might know that. So. Yeah. Yeah, but no. There is a dot there. There is a dot there. So Yeah, precisely if you look at it, then there should be a dot there. So I didn't, so I thought that. Yeah. Okay. Yeah, so then I will go with misprint. All right. Yes. Yes, so there is a dot there actually, so that's an important point. So then we sort of made some assumptions that these nephrons, they are not of same size, and therefore the tubes and capillaries, they will be of different sizes, and therefore the filtration rate would vary from one nephron to another nephrons. And at the same time, since we are focusing on these solutes like protein molecules, glucose, sodium, so it's always important to know how they are moving. I mean, how their concentration is changing throughout the kidney or throughout our body. And since we know that we have 2 million nephrons in our body, we can consider, let's say, scaling parameter as 10 to the power minus 6 when we want to come to the one nephron at the microscopic level. So based on that, we wrote down the equation for the solutes, where we still, this f of u epsilon is the source term, which actually signifies that when we are getting all the sodium or glucose that are being reabsorbed, I mean, this f of u epsilon will actually denote that sink term in your equation. And this u epsilon can be sodium, glucose, or protein, or any other molecule, any other solute in the body. And that's the equation. Concentration of solutes, yes. And then we also need to know how the blood is flowing. So the q epsilon denotes the velocity of the blood, and since blood is a non-Newtonian fluid, we can model it using Navier-Stokes equation, which is given by this way, where P epsilon is the hydrostatic pressure, and F epsilon can be the shear stresses, shear stress in the blood. And since this pressure is actually getting fed in to all these calculations, so whatever P epsilon will calculate after doing the homogenization, the same P epsilon will, P0, whatever P0 will get after the homogenization, the same P0 must be plugged in to know the overall. Yes, so we are basically considering one single nephron, and in that, let me go back. I have to go back a lot of slides. Okay, so
Just say what is epsilon.
Epsilon is the scaling parameter for one I mean, when you scale down everything to epsilon scale, so basically we are now focusing on that epsilon y scale. So like, which equation? Yes, yes. Okay. Oh. See, sometimes we actually saying the same thing, but it's just that we are, I mean, I forgot to use the same term or something, or I mean, yeah. All right. Yeah, and then since the pressure from here will fed into these equations, based on that P epsilon, we can be able to calculate the physiological changes in the kidney. But in order to address this micro-macro model, there are several issues. So first of all, we don't know how the reabsorption is taking place, whether we can write it as a linear function or a quadratic function, or simply if it is a Lipschitz function, or I mean, so far we need to know how this reabsorption rate can be taken. So that was the first issue. Second one is, so I think I have a wrong spelling of scaling. So I admit it by myself. So we don't know whether the scaling is done correctly or not. So we can still play with the scaling. Another problem is we also need some boundary conditions to complement this entire model. And the fourth issue is whether we can get some a priori estimates or not, because these two equations look simpler, but the rest of the equations might create some problem. So we need to have some kind of a priori estimate for the two-scale convergence, and based on which our goal would be to upscale this entire model. And as a mathematician, I would be interested to know whether we have an existence of solution, whether we can apply the linear theory of parabolic equation or whatever fixed point arguments to show the existence of solution or not. So that will be the next goal in this direction. And since we got the grant, I believe last year, end of December, so far we don't have any results to show, so we are still working on it, and hopefully we'll have something in coming months. And to conclude this second model, our model can predict the physiological changes in humans if we have diabetes. And some suggestions were from clinical team, which you hear all the time from the doctors, from relatives, from your well-wishers, that exercise regularly, lose weight, healthy food choices, low salt diet, don't eat a lot of medicines. Yeah. No. Yeah. Yeah. Yeah, exactly, yeah. Yeah.
Okay. And let me ask you, is it some confidential thing here?
Yes. So I'm not allowed to.
So you are hidden almost everything.
So I'm not allowed to release any kind of. Yeah. So I'm not allowed to release any kind of experimental data or any. Yeah. So
you are only talking about the.
Exactly.
Anything about the domain. You are not shown what kind of.
So right now, this homogenization part is still work in progress. Yeah.
You are not showing what kind of domain to study the existence and things like that. You need to have a proper domain.
Exactly.
It's not like going.
Yeah. Well, that's the detailed homogenization part. Yeah. That's where we are currently at the moment. Yeah. Yeah.